just make sure that you understand the order of the enzymes: helicase, primase, DNA polymerase 3, DNA polymerase 1, and then DNA ligase. Helicase attaches to origin of replication, unzips DNA, breaks the hydrogen bonds. Primase has to come and put a short, this is actually transcription. So helicase attaches to origin of replication, unzips DNA, then primase comes, has to read 3 to 5, put a short RNA called primer, so the 3' carbon would be available for DNA polymerase 3. The DNA polymerase 3 attaches to the end of the primer, reads 3 to 5, and puts complementary nucleotides. Primers have to be removed. RNA can't be part of the DNA. Therefore, that's the job of DNA polymerase 1, comes and removes primers. And finally, there are gaps in the sugar phosphate. Ligase takes care of that. Replication happens in 2 forms for the strands. One is called, the new strand is called leading. Why? Because the direction of replication follows helicase. So it starts with 1 primer, and after that it's continuous. The other strand is called lagging because the direction of replication is the opposite of helicase. Therefore, multiple primers have to be put down. Multiple DNA polymerase jump is fragmented, creates Okazaki fragment. At the end, everything is made, semi-conservative. Every new strand, every new DNA has 1 old strand, 1 new strand, and so on and so forth. Then we started talking about protein synthesis or gene expression. So like I said, forget everything you learned about DNA replication. This is a different process. The things that are involved, the characters that are involved in gene expression are the genes, which have a promoter region and a terminator region. So every gene has a beginning. Every gene has a beginning and an end. That's called the promoter, the terminator sequence. All right? Then you have 1 single enzyme that is responsible for the first part, which is transcription. That's called RNA polymerase. The second part is translation, where after you make mRNA, you will translate it with the help of ribosomes and tRNAs. Okay? So those are basically what are involved in the process. The gene, RNA polymerase, mRNA, ribosomes, and tRNA. Okay? Gene expression has 2 steps. Transcription. You transcribe the gene. You make a complementary mRNA. The message is put in a single-stranded RNA, which is messenger RNA or mRNA. Transcription starts when the promoter is active. Again, I said don't worry about the gene regulation process. You don't need to know it for this class. Promoter becomes active. RNA polymerase attaches to it. This is the only enzyme that is involved, does it all. Attaches to the promoter, unzips DNA, chooses 1 single strand as a template on DNA, and that will be the strand that goes from promoter to T in what direction? How do we read DNA?
3 to 5.
3 to 5. So it will be the 1. So again, whichever, if I choose this one 3 to 5, then it will choose this strand, vice versa. So the strand, it will read that one and will make a complementary RNA for it, single-stranded, and that's called mRNA. That's the end of the process of transcription. Okay? Very simple. So far, so good? All right. So, now, gene expression in eukaryotes and prokaryotes are technically pretty much the same, except with some minor differences. And that has to do, again, with the setup of the cell. the structure of the cells, the differences between a prokaryote and a eukaryote. So I'm going to put that here. So a prokaryote versus a eukaryote. All right? First, the process of transcription. For both of them, location is in, not both of them. For the prokaryote, the location is cytoplasm. That's where their circular DNA is. What about for us? Where is transcription happening? Where are our chromosomes in our cells? Nucleus. So transcription for us happens in the nucleus. During the process of transcription, the mRNA is made. This mRNA is ready for translation. In a prokaryote, this mRNA is ready for translation. For us, it is not. It needs to be processed further. That again is because our system is much more complex. So again, that's the process of transcription. Remember, I have two different animations. Whatever works for you, use that one. For us, our mRNA needs to be processed. mRNA has to be processed. Sorry for my fast handwriting. How does it need to be processed? Three things happen to it. First, introns are removed. All right, so what the heck is an intron? They believe where introns came from was the process of evolution. And as eukaryotes evolved, what is the difference between one species or the other species who are evolutionarily related to one another is in their genetic makeup. Our closest relative are chimpanzees. Our DNA difference is less than 2% differences. Many of our genes are identical, but a chimpanzee is a chimpanzee. That 2% difference makes them chimpanzees, makes us human. We have a specific gene that allows us to speak. They can make sound, but they cannot vocalize. It makes our larynx much better and our brain to be able to process different sounds and up and down and all of that. But they don't. And so other differences that makes them chimpanzees. But seriously, the rest is identical. So they think evolution happens because genes change different sequences and all of that. In eukaryotic organisms, when they started studying proteins and they looked at the proteins and then they said, okay, let's create an imaginary mRNA for it, and they extracted the mRNAs from the cytoplasm and they sequenced it, then they looked at the genome. Human genome has been completely sequenced. When they looked at the mRNA and changed that into what the gene sequence should be in DNA language, then they looked at the actual gene. The gene was way longer, whereas mRNA was way shorter, and there was some pieces that were missing. What that is are introns. So within our DNA makeup, even in between the genes, there are sequences that now have become silenced. We call them junk DNA or introns. And they think that's evolution. So in order to change the protein, rather than rewriting the whole DNA, they masked, they said, okay, silence this, the new protein looks like that. All right? So we have introns. You cannot make a functioning protein in humans if you have the whole introns in there. The cell knows the location of the introns. So when the mRNA is made in the nucleus, there are certain enzymes that come and go through that premature mRNA, find the introns, and they can actually loop and excise or remove the introns. This is another image of it. So it basically goes and the enzyme attaches, loops the DNA, kind of brings the two ends together, cuts it, and sticks them together. Kind of like how editing was in the old days when we didn't have digital. We had the movies, right, the film. I don't know if you've ever used the projector, but what they wanted to do is they wanted to take a scene out. So the director would go to the editor and say, I want this whole entire scene out. So the editor would look on the film and say, okay, the scene starts here. We'll cut it there, and the scene ends there. We'll cut it there, and then we'll tape them together. That's how Intron is working. Or if you've ever seen the old projectors, when they break, all you have to do is cut them and stick them together.
So basically the introns are like the tape that connects
No, intron is the piece of the film. It's the scene that you take out. It's junk. You don't want to use it. The parts of DNA that have the sequence are called exons. So you stick the exons together. Exon 1, you take the intron out of it, stick it to exon 2. Same thing, all right? Now you might say, why did they name it like that? Introns makes me think like it's in, exon is out. Think of what comes out of the nucleus and is used. Exon pieces are the pieces that are used that exit the nucleus. That's why they named it that way.
That technically be the mRNA, so.
Yes, so they will split it, stick it together, and that's your mature, semi-mature mRNA. One, you have just exons. All right, that's the one that is going to be translated. Everyone okay with introns and exons? Next thing that has to happen is you have to add a 5' cap. That's so ribosomes can attach to it. Don't worry about it, just say a 5' cap. It's a sequence of mRNA, sequence of RNA that they attach to the tip. It's consistent for all cells, all right? And for every cell, for every mRNA. Ribosomes know. And finally, at the end of it, they have to add a poly A 3' tail. So multiple A RNA nucleotides are added to them. Once that happens, this is the mature mRNA. All right, so. So I will choose the 3' to 5', right? And let's create a silly sequence, whatever. Here, I'm creating, all right? So I'm going to read that and make my mRNA. mRNA is going to be off of this, complementary to it, and anti-parallel. So your mRNA will always start at 5'. So I'm going to see in A, they have to put a U in there, a T, and A, G, G, C, U, U, A, all right? And continue. My mRNA's order is this way, 5' to 3'. So 5' is the start of your mRNA. And we have to, for Eukaryotes, we have to add a 5' cap. That's where ribosome starts reading your mRNA. So for reading mRNA, it's the reverse. You read mRNA 5 to 3. Why? Because your DNA was 3 to 5. So if 3 is the start of your gene, 5 has to be the start of your mRNA because it's complementary to it, an anti-parallel. Everybody understand what I'm trying to say? Okay. It's hard to put it in words. All right. Good. We're done with transcription in eukaryotes. In prokaryotes, the second RNA polymerase fell off of the T, mRNA was ready to go to translation. But for us in eukaryotes, take the introns out, add a 5' cap so ribosome attaches to that. Then add a poly A tail. Now your mRNA comes out of the nucleus and is ready to be translated. So translation process. For them is in the cytoplasm. For us is in the cytoplasm also. But our ribosomes are found in 2 areas. So either cytoplasm, it could be just free ribosomes, or in rough ER, right? That's where our ribosomes are. But technically, both of them are in the cytoplasm, so technically translation happens in the cytoplasm. The process of translation for us is exactly the same, okay? There's one other difference that I'll mention it here as we go through. All right. So translation, again, let me remind you about how DNA speaks to us. DNA has nucleotides. So it will speak to us in nucleotide language. The words that it creates are called codons. A codon is made up of 3 nucleotides. 3 nucleotides together make a codon. So the message is on mRNA. Therefore, we didn't go and translate in DNA language because ribosomes read mRNA. So codons for us are in RNA language. Same thing for prokaryotes. So codons will never have a T, they will always have a U because they are in RNA language. Codons are found on messenger RNA. A codon is one specific amino acid. The codons that we have discovered, there are 64 codons, 64 possibilities. We refer to this as redundant genetic code. Redundant because we only have 20 amino acids. So therefore, for the cell, in order to make sure all the 64 possibilities have been taken care of, for some amino acids, they use multiple different codons. UUA means leucine, UUG means leucine, CUU, leucine, CUC is still leucine, CUA is still leucine, CUG is still leucine. So, it's kind of like I said, hey, hi, hello, yo, all these different ways, good morning, good afternoon. These are all greetings. That's how you welcome someone and you greet them. Different words immediately in your brain brings the same reaction. Somebody is greeting you, all right? Aloha is greeting or goodbye. That one is weird. All right, so they mean the same thing. You also have to tell the poor ribosome, okay, where do you start with the codon? Because we've added, even look at us, we've added a 5' cap. So when the RNA polymerase attaches to the promoter, it doesn't care where is the start, where is the end. It just knows, okay, I'm gonna attach the promoter, unzip, and read whatever I read and put it in RNA language. It has so much to do, it doesn't care where introns are, where it starts and all of that. It doesn't matter to it. It will just say, listen, you've given me this, I'm gonna go from here to here and I'm gonna put everything down. Then it's up to you to decide. Cells actually have thought about that too. And for every messenger RNA, the first codon, whether it's prokaryote or eukaryote, the first codon is AUG. AUG in every cell means start and put down methionine. Okay. For prokaryotes, AUG means start, put down F-methionine. So that's one of the differences. For them, AUG means F-methionine. For us, AUG means methionine. So the ribosome knows, all right, when I attach the 5' end of this guy, I have to go down and find AUG. From that point on, this is my first codon. The other thing that has to be related to ribosomes is where is the end of it? Again, for us, remember, we add a poly A 3' tail to the mRNA. So we add a cap here and bunch of A's to the bottom. So where do you stop reading it? For that, the cell has also come up and said, all right, these 3 words mean stop. And that's all they mean, stop. They will not mean any amino acids. And that's actually how ribosome knows, oh, this is the end of it. No amino acid is coming. Instead, for those, there is a specific protein that sticks there, and that's when the ribosome sees that, goes, this is the end, and separates. So, and again, you don't have to memorize it. UAA, UAG, and UGA, all 3 of them means stop. So technically,